All right, good afternoon, everyone. It was kind of cool talking to students after the lecture because there were multiple cases where people have encountered this kind of stuff. Using a chemical to cause a biological effect is a really broad area. And so I do want to encourage you as you look at this, find the connections to what you've done, the things that you've thought about, the jobs that you've done. And I'm trying to provide enough detail where you can make those connections and ask those questions. As for what's on the test, we'll talk about that a little bit during the review session. But again, not every little bit of detail is going to be on the test. Instead, the general principles and the kind of questions that were asked in the homework, that's the kind of thing that's on the test. Now, I say that especially right now because as we move into the final part of this lecture, we have a lot of things to talk about, but we're going to try to find the similarities between them. And our question is, okay, we've talked about how cells sense chemicals. How do cells sense light? And it turns out the way they sense light is connected to the way that you have two other senses, the sense of smell and the sense of taste. So that's what we have right there. It all starts with the eye, and as biochemists we are interested in what happens inside the retina as the light comes in through the lens, hitting the retina, and sending a signal down the nerves to the brain. And it turns out that this as well depends on a serpentine receptor and a G protein. So we have the same principles. In fact, if you think about this, this is kind of weird. Why should a membrane protein that's for sensing things that were outside be used for sensing something like light that can actually go through into the cell? And in fact, this membrane that these proteins are in is actually located inside the cell. It's not the extracellular membrane, it's little membrane discs inside the rod and cone cells. All of this means that it looks like it's an evolutionary vestige that this was a membrane protein that was working with light in bacteria, and then we adapted it for use in the eyes. It's really kind of cool that the same kind of setup that we use for adrenaline is also used for light. The one difference is, of course, you're not binding something from outside the protein. You are you're binding light. The way that you do that is you have a light-sensitive molecule that is buried deep within the rhodopsin. It's called retinal, and if you see that blue structure, that is what the retinal is. Now down below that you have transducin, which is a G protein. In this case, the G protein is arranged in alpha, beta, gamma, but we call it transducin because it's special. But otherwise, if you look at the structure, it's definitely following the same rules that we've been talking about so long. So this is an example of the neurons that are connected to an eye. This is not a human eye. This is a daddy Longlegs eye. But the green lenses focus the light onto the rod cells. In this case, they are blue and purple. And you can see that they are all arranged so that the light falls upon them, and then they send signals back to the brain. So we're talking about that initial event. So a Drosophila eye, you have here the retina being shown, you had the photoaxons in blue, and then you had the brain neurons in green. And this all makes sense. You have the retina on the outside, and then you have the nerves behind the retina. Unfortunately, that's not the case for us mammals. For some weird evolutionary reason, we actually have the eye reversed on this one point. If you notice that it has the rod and cone photoreceptors in red on the inside when the light's coming in from the upper left corner here. So it's actually, the light has to pass through the ganglion cells and the amacrine cells and the bipolar cells, and even I don't know what those cells mean. What I just know is the light actually passes through several layers of cells before it gets to the photoreceptors in this case. And then it comes back out, does a U-turn, and goes through the optic nerve to the brain. That's just the way it is. I can't completely explain why, but I can also say maybe it doesn't need to be that efficient on that point, because on the points that matter, it's incredibly efficient. It's efficient to the point that a single photon can cause the firing of the nerves, which sends a signal to the brain. You can't get any more sensitive than that. And so maybe the sensitivity of having it backwards like this just doesn't matter. So your book does show it like this. We aren't interested in the different kinds of nerves so much as we are interested in how does the signal get started. One of the topics that we can't study this year is the question of how does the signal propagate through a nerve itself. We're just going to deal with the first step, and the later steps will be dealt with in other classes. So rods and cones are vision cells, and you see that they have these discs of membrane, and they have all those membranes that hold the rhodopsin, or the opsins that they use to gather light. The rods and cones have very different shapes, but inside they're pretty much the same. There's only a slight difference in their proteins that they use in their opsins. So there's lots of membranes, and then you have a polarization which happens due to changing the voltage by letting ions in or blocking ions from coming in. Either one of those will change the voltage and will start a voltage potential that can then travel to the brain. So our question right now is, how is this electrical impulse started? You can tell if there's going to be a voltage change that we're going to have some kind of ion channel opening or closing. And that's what we talked about way back when we talked about section 11.3 back in chapter 6. So here's the mouse showing the nerve fiber layer. These are very busy nerves, and in fact, you could argue that the busiest nerve in the body is the optic nerve, I believe. There's lots of tumors that happen there as a result, a lot of optic nerve tumors, because there's so much activity. You get reactive oxygen species, you get DNA damage, and you get cancer as a result. So these are doing a lot of things because the vision is very important to us. And so this is what it looks like in your book. I mean, this is again a figure where I'm cutting off the top of it to show you the parts that are important, and I want you to see the parts that are important are the parts that look like the previous stuff that we've been talking about in the class. You've got the red opsin, it's a serpentine receptor in this case, it's just called rhodopsin, and then you have the green G protein, which is transducin or T. So what you have is you have the same kind of deal where you have GTP-binding, it activates and it goes to an enzyme that is blue, just like adenylyl cyclase. But notice that this enzyme is not a cyclase, it's a phosphodiesterase. And it doesn't work on CAMP, but it breaks down CGMP. So it's the same but opposite, okay? So it actually goes and it activates by removing an inhibitory subunit. The main thing is I want you to see there's another blue enzyme, but this blue enzyme breaks down CGMP, and when you break down CGMP, you lower the concentrations of that molecule, and that molecule is way on step 5, all the way over. That molecule is the green sphere keeping the purple ion channel open. What this means is when a photon hits the eye, CGMP goes down and the channels close. So you naturally have in vision cells, you naturally keep a sodium-calcium channel open. And it's letting sodium-calcium, it's like keeping the window open on a cold day, or keeping the door open. It's literally a door for ions, right? And it's keeping it open on a cold day as the sodium-potassium ATPase is constantly pumping the sodium out. But you keep the door open normally until a photon comes in, and then you close the door. And then the voltage goes up. The difference between inside and outside goes up because you have the pump working and you don't have the door open anymore. The membrane becomes hyperpolarized. And that's because of the constant action of the ATPase. Just remember that that is central to how all this works. Then you have a hyperpolarization and the signal passes through the brain that way. This is a keeping out the calcium-sodium second messenger, in a sense, that is coming in from outside because it's being transported. All of these things have parallels to what we've talked about before, and that's what's important here. So I just want you to look and see how that, changing the closure or open state of an ion channel is the way that light is sensed. Now the bottom of this, it gets complicated. And so you have another, this is the way that it's turned off, because you see it's the recovery adaptation phase. And honestly, we aren't going to talk about this at all. Just to say that it has some of the elements that we've seen before, including there is a specific arrestin that will take the rhodopsin and it will bind it and pull it to a place where it's no longer active. And that's how your eye cells turn off the signal. All the other stuff, don't worry that much about it. You just reverse the stuff that went before. That's too much detail. Again, we're going to stop once the signal is sent. So the main thing here is this actually shows the sodium-potassium ATPase. And so the sodium-potassium ATPase means that you have the cGMP. When the cGMP is taken away, the door is closed, you move to the left, you move from the left to the right, and the voltage goes up from 45 millivolts negative when you start to 75 millivolts, because you no longer have the sodium and calcium flowing in down their concentration gradient. So vision is the champion of signal amplification. I think that this weird hyperpolarization arrangement that they do is one of the reasons why it works. So you can't get much more optimized than one photon equals one excited rhodopsin molecule equals a nerve firing. And you can actually get to that. The reason why this works is because one rhodopsin can create 500 transducins, which can activate 500 phosphodiesterases, and those can work really fast and amplify. Those can do 4,200 GMP a second. And that's a lot of GMP that can be chewed up, or a lot of GMP product that can be produced from cyclic GMP in order to create a signal. Basically, a single photon can result in thousands of ion channels closed, and that can cause a membrane potential change on the range of, something that can kickstart a nerve into actually sending a signal to the brain. So that's how photons are sensed in general. That's how rod cells work. They just sense whether there's photons or not. Cone cells are different because each cone cell, there's 3 types. There's red, green, and blue. And each type has a slightly different opsin. And all this is it's the same protein, same retinal in the middle. But there's different amino acids around the retinal so that it absorbs at a slightly different place. The rhodopsin absorbs light in the middle of the visible range, and that's just because it just wants to absorb light. But the green, blue, and red has been shifted out of the middle of the range to the edges of the range. And that allows you to sense whether it's close to the edge, or whether it's close to this edge or that edge. And that is where you get different colors of light sensing from. So just like computer monitors have red, green, and blue pixels, in the same way your eye has red, green, and blue sensing cells. They go together. So in fact, there's kind of a cool story about one of my favorite chemists, John Dalton. And he was very important. Of course, he gave his name to the unit of Daltons because he was really important in us figuring out that atoms are even where it's at when it comes to chemistry. He was one of the first true chemists. And he was also colorblind. And so he was very curious about this. His hypothesis was the fluid inside his eye was discolored, which actually was a reasonable hypothesis. Of course, he didn't want to test it at the time because that would require sacrificing your eye to science. But he did put in his will that when he died, he wanted people to open his eyes and look inside and see if the fluid was discolored. Well, of course, it was clear. His theory was wrong. So John Dalton's last theory was wrong. I don't think he minds because now we know why he was colorblind. And you can actually still see the eyes. They were preserved in formaldehyde. So these are John Dalton's eyes that are in a jar somewhere, probably in the UK or something like that. It seems like that's where they would have it. So because they were preserved, we actually were able to do PCR on them with and figure out the DNA. We could look at his genes, and we looked at his opsin genes. And it turns out you have a different gene for blue opsin, red, and green. And he did not have a functional green opsin gene. That gene was messed up. And so after much later, more than 100 years after he first wondered about this, we did actually have proof of what his problem was. And so there's lots of interesting things that can happen with the eye. In fact, if you think about it, if you can just shift the opsin a little bit and do red, green, and blue, is it possible to shift it a little more and maybe have something that that absorbs ultraviolet light and causes the eye to fire for that? In fact, not only could this happen, it has happened. There are certain kinds of cataract surgery that give you the ability to see UV light. I think it messes with your lens rather than the opsins. But whatever is the cause, the result is your blue opsin, I believe, gets triggered by shorter wavelength light, more UV light. And it would be like you're looking around the world and you see it's lit up by a black light. And Monet actually captured this in some of his paintings. Some people think that this is why his paintings have sort of an ultraviolet feel to them. It's a little bit like what he would see. He physically saw these blue tones, these violet tones. There's a little bit at the bottom about forbidden colors, but that gets a little bit more complicated, and that has to do with signaling. You can talk about that if you talk about the nerve signaling in another class, I suppose. Let's just focus on Claude Monet's UV vision for now. So if you can shift if you can just shift the wavelengths, maybe there's ways to do that with chemistry. And in fact, there are nanoparticles that will actually give mice the ability to see in infrared. So this is the other end of the spectrum. This is longer wavelength rather than shorter, but you can just have nanoparticles that will absorb in the infrared and they'll give off in wavelengths that mice can see. So this is kind of like a transmitter. They're literally absorbing one kind of light and emitting a visible light. And there's a group at University of Massachusetts Medical School that basically took these nanoparticles and they gave them, they put them into the mouse's eyes, and it did work exactly like it should. It basically transformed the light into a wavelength they could see, and therefore if they had infrared wavelengths, they could see in infrared. They absorb light at 980 and they emit it at a shorter wavelength, which is an unusual kind of thing. So but the other thing that you might be thinking of is you might be thinking about gene therapy. We talked about that for sickle cell, and there is possibly gene therapy for missing opsins as well as for missing hemoglobin chains or messed up hemoglobin chains. Here's an actual article about this where people actually gave a cone opsin and they actually were able to restore patterned vision, a low degree of vision, to and I think this is in a mouse model. So again, we're healing mice. I don't know how far away this is from humans. But it is an injection of gene therapy viruses that will go to the cells and put the correct gene into the cells that are lacking it. And just if you want to know more about gene therapy, you can look into it. You can look up this stuff. And the thing is, it's actually a pretty simple gene therapy system, and it seems to work really well on the mice. Now, on the other hand, this is a couple of years old, and I have not heard about whether this is going anywhere since this. So, very promising result in mice, but is gene therapy around the corner for color blindness, or is it just too expensive to be worth it? Maybe that's the case, but at least for mice, it actually works as simply as you might think it would. You just got to get the gene into the cells to fix the broken opsin. So that's how sight works. Smell and taste also work with similar systems. And so again, we're going to have the same red, green, blue code. Now, here it gets a little bit complicated because the ion channels are different for each one, but we aren't talking that much about ion channels. We're focusing on the red, green, blue here. You have a red odorant receptor that binds to odorant molecules, and that causes a G olfactory, which will bind GTP, will become activated, and that one will go to an adrenalyl cyclase, which works just like the adrenaline system. That cAMP will go, and that will actually open some channels, and it will those will cause other ones to let chloride out. That gets more complicated down the road, but I want you to focus on the steps up to the creation of cAMP. that works that way. Very similar to vision neurons. And once you have the system set up, again, you can have a trigger of smell that can go to the smell part of the brain, and that can send a smell signal. This is where Linda Buck works, and she actually had the lab next door to mine when I was at the Hutch. I think she's still at the Hutch. And she came to the Hutch when I was there as a postdoc. And at first I was like, why is a smell researcher coming to the Fred Hutchinson Cancer Research Center? And then I learned a little bit more about how these things work, and I learned that RAS, the cancer protein, is in the same chapter as G proteins, which work with smell. And so it turns out that signaling is actually very closely related to cancer. So there's a lot of work for Linda Buck to do. A Nobel Prize for smell fits right in at a chemistry research, at a cancer research center. And in fact, if you want to know a little bit more about it, there was a whole article in the New York Times about COVID-19 and the mysteries of smell. This doesn't have that much biochemical detail in it, so I'm not assigning it or anything, but it's for if you're interested in making those connections. It has a few connections to make. In fact, one of the connections it makes is it talks about the people who've really figured out what smell is. And look, there she is. There's Linda Buck, who's basically the world expert in figuring out how smell is perceived. Taste is a lot like smell. This makes sense because they're very closely related senses. Taste has a sensory receptor that binds a tastant. So like the sweet receptor will bind sweet molecules. And then you have, again, G-protein, adenylyl cyclase, and this one actually goes to protein kinase A, and then it goes and opens and closes different things. So there's another thing that we've seen before, but this looks even more like the stuff that we've talked about before. First messenger, second messenger, G-protein, kinase, and a cyclase. They're all involved here. So that's what the G proteins are that we'll talk about. GSs are stimulating, and GIs are their opposites, they're inhibitory. And if you have a problem, we might talk about this a bit on the review day. If you have a problem with an overstimulated G protein GS pathway, then what you want to do is find a GI pathway that you can trigger that will turn that GS pathway off. Inhibitory G proteins are actually very helpful. Transducin is a G protein, even though it doesn't have a G in it. G-olf and G-gust are for olfaction and gustation, which are smell and taste. So all this is put nicely together in this summary protein, which shows you kind of what you need to know, especially the top half of this figure. And it even shows you like an odorant pathway that we don't talk about, the inositol phosphate pathway. Don't worry about that one. But look at all of these where you have a GI vasopressin, you have a GS epinephrine, and those both signal through pKa, but they have the opposite effects on cAMP. So that makes perfect sense. Light works in sort of a reversed way by a phosphodiesterase, and vision is sort of flipped over in that way. But odorants and tastants work through cyclases just like the other ones. So see all the commonalities in this picture, and don't sweat too much about the bottom ion channel parts of it. So if you go back to insulin receptor, you actually have a glucagon that is a anti-insulin. This is an inhibitor of the insulin receptor, and it sends the message release glucose from storage. Now, glucagon actually is a G protein. It actually works through a GS pathway, and it has cAMP as its second messenger. So that shows you that these different kinds of pathways can sometimes work together. Glucagon acts like a GI to anti-insulin, but because insulin itself is not a GI or a GS kind of pathway, it instead uses a GS pathway and it wires it differently. It wires it so that it turns off the insulin pathway. And I wonder why this is. I think that it basically might be something, the insulin pathway causes so many different signals that probably it's why you have the weird thing where it's being turned off by a GS pathway, even though it itself is not a G protein pathway, it's a tyrosine kinase pathway. It's important to know what glucagon is because it's the opposite of insulin, and it's going to come up in Biochem 2 quite a bit. Oh, and I do have this. This is cool because we were just talking about this. So cholera actually causes a dysregulation of a G protein pathway. Pertussis does the same thing. It just does it for a different kind of G protein. So you can have very different effects, but you can have these toxins that target G proteins. So this is the way cholera works. It actually takes an NAD molecule, which you can see the adenine in the box over there. And what it does is it takes this NAD molecule and it forms a covalent bond to the GS that goes to adenylyl cyclase. Once you put this adenylation group onto the arginine of the GS, then it actually will be ADP ribosylated. So you put an ADP onto it. What happens is that prevents it from ever breaking down its GTPase, which means that the adenylyl cyclase is constantly on, constantly poking out CAMP, and constantly causing chloride to fire from those cells, which causes a water imbalance, which means that basically water is gushing out of that organ. When this happens in the gut, you have cholera and you have diarrhea. When this happens in other places, I was just talking with one student about it, this happens in other places, it can probably cause a similar kind of water release from that place. And that's why you might stimulate a GS with adrenaline to cause water to be ejected. Cholera does that same trick covalently. And the problem with cholera is you can't turn the faucet off. And then the people die from basically ejecting so much water. The good news is it's relatively easy to treat cholera. That's why we don't have a problem with it in our country, because if you just can hydrate them until the storm tides over, eventually you replace those proteins and eventually the cells heal. But if you can't do that, then you can actually die from it. The end result is this overproduction of CAMP, which causes an overproduction of other things, including chloride and water. So salty water is dysregulated and leaves the organism too fast. So the cool thing about all of this, the whole thing about G protein coupled receptors is that they're modular, right? That's why we had that one thing that showed all the different G's on it and how they were all the same color, red, green, blue. And so because they're modular and because evolution has traded them out and made different signaling pathways, well, maybe we can do the same thing. And here's some people that did that with yeast. They basically wanted to use a yeast G protein coupled pathway, and they wanted to switch out the G protein so that the yeast would be activated by whatever they wanted. And so they basically put in different receptors into the yeast, and they made yeast that were reactive to make a different color for different molecules that came in. And the reason why this works is because you can switch out the different receptors for different peptides or that come from different pathogens. And so then you can make something, I think, let's see, they got fungal receptors for sensitive response to mating peptides secreted by pathogenic fungi. The main thing is they got 3 very different receptors, and they just plugged each of them into different yeast, and they made a different biosensor as a result. The rest of the pathway they could keep the same, and they're just trading out the receptor. And this makes something that when it detects the fungus, it will turn colors. And so you have a very sensitive and specific kind of detection technique that will work in the field in pretty much anywhere. So I just think that's really cool biotechnology. Now, of course, this chapter is sort of a different chapter. I've talked throughout it about how the homework is going to be a little different. Once in a while, you should look up something in the book. Homework is designed to be done open book. But of course, the tests are closed book. The homework will be, if an idea is reinforced on the homework, then I will be more likely to include it on the test. We'll talk more about that. We'll have some little goodies about what all this stuff means on Wednesday when we talk about these things. But for now, this is enough time for me to finish up the lecture. Congratulations, you have finished all the new lectures for biochem, and this is Monday afternoon. You have 8 days, a little 7.5, until we are going to be taking the test next Tuesday. Oh, yeah, it's 8 days. Okay, anyways, I can do math or something like that. 7.5. Okay, whatever it is. I'll see you on Tuesday, November 23rd for the test. But before that, please come to class so you can ask questions and we can talk about the test on Wednesday, November 17th. So see you in Bertona 4 then. And until then, this is the time where we're going to tie it all together. We're going to bring it all together in a comprehensive final. And once you can show me that you can solve these biochemical puzzles, I can give you a good grade and you can move on. You can use this stuff in real life. That's my real goal. All right, so see you on Wednesday.